We have a great privilege of welcoming to our podium today a distinguished leader in the field of pharmacology, whose career exemplifies excellence across academia, industry, and scientific stewardship. Dr. Martin Michel is a physician whose expertise spans experimental and clinical pharmacology, developed through rigorous training at leading institutions in Essen, Germany, and San Diego, California. Throughout his career, he has held positions of significant responsibility and influence. He established and directed nephrology and hypertension research laboratory at the University of Essen from 1993 to 2002, subsequently leading the Department of Pharmacology and Pharmacotherapy at the University of Amsterdam for 8 years. His breadth of experience extends to the pharmaceutical industry, where he served as a global head of product and pipeline scientific support at Boehringer Ingelheim. Dr. Martin currently holds the position of Professor of Pharmacology at Johannes Gutenberg University in Mainz, a role he has maintained since 2012, while also serving as a senior partner at the Partnership of the Assessment and Accreditation of Science in Heidelberg. His scientific contributions are remarkable. More than 500 peer-reviewed publications with over 37,000 citations, an extraordinary H index of 97. His research focuses on urogenital and cardiovascular pharmacology areas, where he continues to push the boundaries of our understanding. Beyond his own research, Dr. Martin has been instrumental in advancing the field through his editorial leadership, serving as deputy editor-in-chief of both molecular pharmacology and pharmacological reviews, while contributing to the editorial boards of numerous other pharmacological journals. His distinguished standing in the scientific community is reflected in his selection to the Academia Europea, his designation as Honorary Fellow at the British Pharmacology Society, and his fellowship in the American Society for Pharmacology and Experimental Therapeutics. Please join me in welcoming Dr. Martin Michel.
Well, yeah, thank you very much, David. It's a great pleasure to visit here, and so far it has been an extremely enjoyable visit due to the very kind hospitality by David and his wife, which they got up on Sunday at 2 o'clock in the morning to meet us. That's dedication. And apparently, he must have told you that I'm a good-looking pharmacologist, because I see such an excess of female people in the audience. Anyway, what I want to talk about today emerges out of a big consortium project in Europe, where a dozen major pharmaceutical companies contributed, about 24 academic institutions from all over Europe, and we also had some involvement from the NIH. And I currently, as a person, have the pleasure of heading the guideline committee on good scientific practice for IUPHAR, which is the global umbrella organization of national pharmacological societies. So all of that went into this presentation. So I want to do 3 things in the 45 minutes today. I want to get you a realistic impression of the robustness of published data. And I want to talk about 2 big problems. That is the role of biases and the role of poor understanding and application of statistical analysis. Before getting started, the field has no clear agreed upon definitions, so I want to operationally say how I, for the purpose of this presentation, define things. When I talk about reproducibility, I mean that if you do a study exactly like reported, in theory, we expect to get the same findings. Maybe 1 or 2% less, but the same thing, the same answer to our question. And that I will refer to as reproducibility. When I say robustness, I mean a situation where you do a study where, in broad terms, done as a previous study, but with some minor modifications, maybe a different antibody, maybe a different mouse strain, something like that, where you still expect to get roughly similar answer. An extension of that would be if you do a trial in humans, you would expect that animal research done before, to some degree at least, predicts what you will find in humans. And that's the whole basis of research leading to new medicines. And that I would call translational robustness. For those of you who are very quick thinkers, you will have realized a study can never be robust if it's not reproducible. So generally when you read papers, especially in good journals, we assume whatever is written there is true. Well, our trust in that was greatly shattered when in 2011, colleagues from the Bayer pharmaceutical company had this paper in Nature Review of Drug Discovery, where they said, we looked at 67 studies in 3 therapeutic areas that were of interest to us, that is oncology, women's health, and cardiovascular medicine. And to our own surprise, I should say that's something big pharma companies typically do before engaging in a major project based on published data. They want to see, do we see the same thing if we redo the study? Work that very often doesn't get published, but it's something very common in a pharma company. Because once you start a program, you immediately talk like several million euros or dollars. And they reported at the time that in 2/3 of cases, the republished data and the internal data were inconsistent. That is not to say the original investigators were wrong. It's not to say the Bayer people were wrong. It's just to say when the Bayer people did the same experiment, they found completely different results. And then there was a gray zone, and only 1 in 5 studies, the external and the internal data were consistent. And that's kind of shocking. And that single paper really affected a lot of colleagues around the globe. So a high fraction of non-robust studies was found in many other fields: oncology and cancer biology, systems biology, pharmacogenomics, and the situation seems to be even worse in fields such as psychological and social sciences. The overall estimate is that more than half of all published findings cannot be reproduced. More than half. And that's a shocking number. Interestingly, John Ioannidis, originally from the Greek island of Crete, now being a professor both at Stanford and in Berlin in Germany, in certain ways, based on mathematical modeling, predicted that problem. Although it turned out even worse than he predicted it. So another group looked at that from a different perspective, and they said, based on numbers which already are more than a decade old, so this is an underestimate. In 2013, in the US alone, about 50.1.6 billion were spent on research and development in pharmaceuticals. If about half of data are not reproducible, it means that at least $25 billion are wasted every single year. Last time I looked at my checking account, I wasn't even close. Actually, all of us in the room, for a combined lifetime income, will not get even close, unless the next Elon Musk is in the audience. He charges that per hour. But there are other costs to that. For instance, wasted time and effort. I'm aware of stories from PhD students who were tasked from their supervisor to do a project based on a previous student in the lab, and they couldn't get the basic finding confirmed, and only more than a year later, they realized the whole project is flawed. If at my age, I find that I wasted a year, I consider myself lucky. Because after 40 years in research, wasting 1 year, that's reasonable. If you are a second-year PhD student and realize your entire first year was wasted, now that really is not good. We're talking about, of course, wasted research animals, challenges to issued patents, advancing drugs to clinical trials that were bound to fail, even withdrawal of approved drugs, or inappropriate off-label use of drugs based on flawed animal studies. Here's an example for the latter. Most of you will have heard about ALS, amyotrophic lateral sclerosis, an inherited deadly disease for which today we have absolutely no disease-modifying treatment, except for one very, very rare subform. There's more than, actually, according to this Nature paper from a decade ago, at the time there were already more than 50 compounds which in animal studies extended the lifespan of the animals. And in this field, every lab on the planet uses the same mouse. It's a genetically modified mouse, and everybody uses the same mouse. So, and whether the mouse dies or not is not extremely difficult to detect. And among those 50 compounds, there were many compounds which are clinically available. And on this graph, at this ALS Therapy Development Institute, a nonprofit organization in the US, they retested all these 50 plus compounds, whether they, under very stringent condition, that is, adequately powered, properly randomized, allocation concealed, blinded tests, whether they could confirm the original findings. Turns out, here you see in blue the reported extension of life expectancy for some clinically available compounds, and in black you see what they found with very rigorous studies. None of the compounds made it. Zero. And especially with such a deadly disease, typical lifespan from diagnosis to death is 3 years. That has a really bad impact on patients. Oops. Hello, hello. Starship to Earth? Yeah, okay. Yep. So, to conclude this first part, a really worrisome fraction of findings in experimental life sciences is not robust. More than half is the general consensus. That's a scary idea. Think about the work you are doing. I cannot speak on any specific project, but chances are that half of everything you ever did cannot be reproduced by another scientist, statistically speaking. So what are the root causes? Of course, the first thing coming to mind is fraud, fabricated data, falsified data. We don't have hard numbers on that, but there's a general consensus among those in the field that this accounts for maximum 2% of papers. That's 2% too many. Let's be very clear about that. But that doesn't even get close to the 50+%. A second aspect is a poor description of methods. And most likely you all have experienced in your careers, no matter how early your career is, that you saw an interesting method described in a paper where you said, oh, that could be useful for my project, and then found that with the information in the paper, it was technically impossible to redo the experiment because crucial information was missing. Sometimes as basic as the strain of animal not being mentioned, the identity of the antibody not being mentioned, and things like that. We typically refer to that as technical irreproducibility. If the description in the paper is not of sufficient granularity that you even can understand what the colleagues have done, of course you have no chance of reproducing it. I will not talk about that because the solution is very simple. Write your papers so that method section has all the information a colleague needs to reproduce your data. And for a long time, when we still had printed journals, some of the younger people in the audience may not know what that is, there were restrictions how long the method section could be, especially in high-profile journals. Nowadays, where basically every journal at least allows online supplements, there is no excuse not for providing all the details. Absolutely no excuse, other than being lazy. The big issue people in the field say that really matter are biases in the design, conduct, analysis, and reporting of studies, and the combination of a poor understanding of statistics combined with very poor statistical power. In most experimental systems, you will need sample sizes to have robustness based on statistical power of 30 to 100 per group. I don't know anyone who routinely does that, including myself. But that already is a major issue. And then again, abuse of statistics in multiple forms. I will talk about these 2 points in more detail. But there is a fourth area that's poor generalizability. You all are aware of stroke as a condition. And there was a compound called NXY089 that showed beneficial effects in limiting the damage from stroke in 9 independent studies in rodents. And based on that, a phase 2 clinical study was started in which the compound failed miserably. Not that it missed the p-value threshold, no, it just had no effects. And then people went back and looked at issues, and the compound worked beautifully in the studies that were unblinded, but didn't work in the blinded studies. They worked beautifully in the non-randomized studies, but not in the randomized studies. And stroke is a condition which more or less does not exist in healthy people. Only exception, females with a combination of certain contraceptives and smoking. Otherwise, healthy people don't get strokes. And the compound worked beautifully in animal studies based on healthy animals, and it didn't work in hypertensive animals, which is the main reason leading to stroke. So with hindsight, you can say, yeah, well, this drug didn't have a chance to succeed in the clinics. But that's with hindsight. So let me talk about biases. So we as scientists generally think, hey, we are smarter than the average person, which may or may not be true. I don't want to have a discussion on that point. But generally, the human brain is a poor objective evaluator of information. And I want to run a short experiment with you. The next slide will show you a train from the London Underground entering a station. Leaving a station. Leaving a station. And you will just see it for maybe 2 seconds, and I want you to check, can you see the train leaving the station? Yeah? Here is the movie. Who saw the train leaving the station? Okay. Who did not see it? Okay, only a small number. Well, actually, I lied to you. The train was not leaving the station, it was entering the station. Please look again whether you can see that. Can you see the train entering the station? Well, I lied to you again. Men are such cheaters. What you saw is a flicker picture, just oscillating between 2 states. The train was neither entering nor leaving the station. It's just that with being told it's doing one or the other, you created that expectation and your brain lived up to the expectation. And what may come shocking to you is if we look at scientific data, the same thing happens. And I won't go into details for today's lecture. So what's the big problem with biases? Biases are a systematic error. And they can occur at each level of a project, in the design phase, the conduct, the analysis, and the reporting. According both to Wikipedia and a website maintained by the colleagues at the University of Oxford in England, lists more than 60 types of biases. A big problem with this is they can exist concomitantly, and the biases in the multiple phases of a project tend to exaggerate each other. A general idea, which I cannot back by numbers, but I think makes immediately sense, is that if you are looking at an experimental finding that is huge, like a white blood cell expressing interleukin-2 if you stimulate it with an inflammatory stimulus, where it typically goes up like 20,000-fold. If you have this huge effect and a bias like this, it probably will not have a big impact. But most phenomena we study in biology are not changed by 20,000-fold, but maybe a change by 30%, 50%. At least at the protein level. And then, if your effect is only like this, and your bias is like this, the bias proportionally gets a huge influence on the outcome. Let me give you an example how this becomes relevant in the design phase of a study. Many scientists feel that exploratory study is just second-rate science because it's more like a fishing expedition. And I strongly disagree. So a couple of years ago, we did a systematic review of more than 60 guidelines related to data robustness. Some field-specific, some more general. And one of the top 3 recommendations across all those guidelines was to distinguish between studies that really have been designed to test a pre-specified statistical null hypothesis and all the others. And of course, a statistical null hypothesis always refers to one specific outcome parameter. Some of my students recently, in very recent papers from like just the last 5 years, analyzed in a certain area about 100 papers. Not a single one of those studies identified a primary outcome parameter. In clinical medicine, meanwhile, that's a no-go. The statistical analysis plan for a clinical studies always says, this is our primary outcome parameter, and for this we did a power calculation and a sample size calculation, and that's how we did it. In the experimental scientists, we could do the same, but the vast majority of us does not. We need a null hypothesis for a pre-specified parameter, pre-specification of all key experimental methods, including sample size based on power calculations, on all analytical methods, including statistical methods. And then we get a reliable statement related to statistical significance. If any of these conditions is not met, a study by default becomes exploratory, which means p-values cannot be interpreted at face value. But that's what almost 99% of all studies are. And based on mathematical modeling, you can show that these studies have a lower prior probability of finding something. You could say an exciting finding is an unexpected finding. If I leave my hand from the microphone and it falls down, nobody is surprised. If it falls up, everybody is surprised, because it's highly unlikely. If for some strange phenomenon this would happen, most likely it's a false positives. So the more exciting your finding is, the higher the chance, even with a low p-value, it's false. And this paper of John Ioannidis from 2005 predicts exactly that based on mathematical modeling. I had a chance of talking to him about this paper, and he said, well, I sent this paper to multiple journals, they all rejected it, and finally at one paper, it got accepted, and one of the reviewers said, I really hate this paper, but as hard as I tried, I couldn't find a flaw in the underlying math, so I guess it needs to be accepted. And that's what happened. And then for 6 years, this paper was not cited a single time. And only after the Bayer paper in 2011 came out, people rediscovered the paper, and meanwhile, it's the most cited paper in the journal's history, because it laid all the mathematical foundations why we have these problems. Generally, when many scientists feel that exploratory research is lower rated than hypothesis testing research, I think there's a major misconception. And that is, in order to design a truly hypothesis testing study, we need so much pre-information because otherwise we couldn't even do a power calculation, that hypothesis testing studies inherently are not innovative. Typically, you need already 10 studies in the field to be able to design a robust hypothesis testing study. So all the real innovation comes from exploratory work. But of course, at the end of the day, you need then to test the hypothesis coming out of that under rigorous conditions. So, nothing wrong with exploratory work. It's just that, and that's recommendations in many, many related guidelines, readers need to know what is what. As I said, exploratory studies have a lower probability, leading to what's called a greater false discovery rate. That is a greater fraction of studies yielding a false positive finding despite a low p-value. I want to give you an example for study conduct and why we need randomization and blinding as much as feasible. Especially some of the older colleagues in the room are probably familiar with the Rosenthal effect. Rosenthal was a professor who ran for undergraduate students in neuroscience course. And in neuroscience and behavioral neuroscience, people love maze tests. Like here, where you look, how long does it take the rat to find the cheese? And if the rat finds the cheese quickly, we think it's a smart rat. And if it takes a long time, we think it's a stupid rat. And as part of the undergrad course, Rosenthal wanted to make it a little more interesting for the students. He let them practice on this test. All you needed was a piece of cheese, a rat, and a stopwatch. And to make it a little more interesting, he said, we have bred a substrain of rats that are smart and a substrain that are stupid, and you get a couple of rats from this strain, a couple of rats from that strain. And of course, the students found inevitably that the smart rats found the cheese more quickly than the stupid rats. The big flaw was he had lied to the students. Not the rats, but the students were the guinea pigs. Because it was a study on how easily a student can get biased. The two strains he talked about never existed. They all were just random rats from the colony. But being told, like I told you with the train from the London Underground, it's going this way, then that's what people found. And if you told them the other rats are going that way, that's what people found. When in reality, no effect existed. And that became to what's called the Rosenthal effect, or in more technical terms, investigator bias. The investigator expects a certain finding, then you will get that finding. And in most cases, you fool yourself. Here is something more realistic and more recent. Emily Senner is a junior faculty member at the University of Edinburgh, and that's still work from a PhD project. And she and her colleagues went to a big meeting and systematically looked at all the posters, several hundred posters. And for each poster, they only checked for the main parameter, is the outcome neutral, / negative, or positive? And did they apply randomization? Did they apply blinding? What you can see here, there were more positive than negative findings. Not surprisingly, it's easier to get money from your boss to attend the meeting if you have positive findings. However, the chance for a negative finding was much bigger if you had randomized or if you had blinded. Odds ratio of about 3. If you agree that a blinded randomized study has higher technical quality than those that are not, most likely this excess of positive findings are all false positives. And if they went even further and looked at the studies that had applied randomization and blinding compared to double absence, the odds ratio of a neutral outcome became 5, meaning that about 80% of the studies with positive findings, unblinded, non-randomized, were false positives. That's about what the Bayer paper found. Here's another example of that. And that is stroke study I already mentioned. The dark gray horizontal bar is how much reduction in brain infarct area is expected to be clinically relevant. And that was easily reached in the non-randomized, but not in the randomized studies with hindsight, after the clinical trial had failed. It was easily reached in the non-blinded, but not in the blinded studies, and it was easily reached in normotensive, but not in hypertensive animals. So the biases led to a clinical program, which cost a lot of money, when, at least with hindsight, that compound never had a chance to succeed in people. And the same problems exist in analysis and reporting. When people, for instance, go ahead and they get a borderline p-value of 0.055, and then they maybe you should use a different statistical test. Maybe there is an outlier in there, and then the p-value gets lower. All this are high-risk biases. So biases reduces robustness, but the big problem is it can occur at any level of a study, and they all potentiate each other. A little fun things about statistics. And no, for most of you, using fun and statistics in the same sentence sounds a little awkward. There's general misconceptions about p-values. In any data set, there's a fixed mathematical relationship between effect size, variability, sample size, and the p-value. And we typically attach the p-value to the effect size. But, as I will show you on the next 2 slides, a p-value can be completely driven by variability or sample size. We can use this fixed mathematical relationship in data analysis where we know the sample size, the observed variability, and the effect size, and then calculate the p-value. Or we can use it in forward mode where we assume variability, an effect size, and a p-value, and then can calculate how many samples would we need to have a certain chance of detecting this effect if it existed in reality. These are fake data. I just took 0.97 and 1.03 and then systematically subtracted 0.01, 0.02 and added it in both groups. And this is what you get. So sample size is small. Effect is small. It's about 5%. Mind you, in very few systems in biology, a 5% difference is meaningful. And variability is very low. SD here is about 1%. I've never seen any biological system in my 40 years of science where biological variability consistently had an SD of 1%. I don't think it exists. Maybe it does in particle physics, but not in biology. Nonetheless, under those conditions, we get a p-value of 0.002. Some people may get excited about the low p-value, but in reality, there's no difference. What are you getting excited about? Now, I took the same data and now did not add and subtract 0.01 and 0.02, but 0.1 and 0.11. So still small sample size, 5% effect size, and SD now is about 10%, which generally spoken I've seen systems where this happens, but in most systems, an SD of 15, 20, 30% is more realistic. I don't know how many people in the room, for instance, do PCRs, but we looked at a long series of PCRs done by multiple people, and we find that the SD in a PCR experiment is about 15 to 20%. Anyway, under those conditions, the p-value jumps to 0.4, and nobody would even think any further. I could go one step further by entering each of those numbers 5 times. So artificially bringing sample size to 25. And then, again, I would have, with the very same data, just a sample size of 25, a p-value of 0.0039, so below the magical threshold. In any case, this effect size is irrelevant. But just by changing variability and sample size, I can get whatever I want. So, what does it mean if something is not statistically significant? Actually, no test can prove the null hypothesis. You can only say there is enough evidence to reject the null hypothesis. If the null hypothesis could not be rejected, it means just that. Possible reasons include there is no difference, effect size is too small, sample size is too small, variability is too large, or it's simply based on random chance a false positive, a type 2 error. And all of these can occur at the same time, actually. Power, I already mentioned. 80% power means that we have an 80% chance that if an effect does exist in reality, in biology, or in clinical medicine, to really find that. And that's standard, 80% power is standard for clinical research. Power is driven by effect sizes, sample sizes, and low variability. Well, okay. I don't know how much money you have, but it may be worthwhile investigating in a new device. Anyway, here's a real-life analogy. A child is sent to the basement to find a tool. After a while, she comes back and says, "It's not there." Well, is it or not? It depends. How long did she look? That would be sample size. Is the tool as large as a snow shovel? And I realize in Mexico City you don't use snow shovels a lot. Although I saw some snow on one of the mountain caps yesterday. And is the basement a real mess like this one, or very well organized? Here's another cartoon of that. On this slide, 6 scientists are giving a small sample to identify what the real object is. And based on the small sample, they come to very different conclusions. It's a spear, a fan, a wall, a rope, a tree, or a snake. For those of you who didn't realize it yet, the true answer would have been it's an elephant. But based on a small sample size, based on chance alone, none of these otherwise highly esteemed colleagues detected that. Here's a real-life experiment. When I ran one of my statistics courses, together with the participants, we designed an educational tool on statistics. And each student picked 2 sets of 10 samples from a real database of real patient data. Since they all were picked from the same database, the null hypothesis was true because there were no 2 groups, just artificially picked. And The true mean of this database is about 13 point something. With a sample size of n is 10, which probably is bigger than what most of you use on a routine basis. They got values for group A ranging from 10 to 18, here in group B from 12 to 18, and in some pairs the second group value was smaller, in some it was larger. By average, across all 200 values, the real difference was close to 0, as it should be, but that was with n is 200. With it n is 10 for each group, all kinds of effects could be seen, and in some cases with a p-value as low as 0.002. None of this is real. There was no difference, although many would observe difference. As this was from a real clinical database, I can tell you in this clinical condition, based on meta-analysis of dozens of studies, the difference between placebo and most effective treatment is around 0.8. So almost everybody found a greater difference than what is clinically achievable. So all of them would have been kind of excited, especially the one that had a very low p-value. None of this is real. So underpowered studies provide biased results because this person finding this huge effect with this low p-value most likely would go ahead and publish it very, very quickly, whereas this person finding more or less no effect, or this person finding it gets worse with a p-value somewhere here, probably also wouldn't publish. So the literature would say, without any contradiction, this new treatment works wonderfully. And it doesn't. Hello? Okay, so statistical power, big sample sizes, are essential for robust conclusions. And this is an analysis by Catherine Button, and she looked at meta-analysis published in 2011 in Neurosciences, and based on the meta-analysis, she could back calculate for every study going into them, said what is the true effect size and what is the true variability, and then checking, based on sample sizes, how much power did the original studies have. Only 1 in 7 studies had a power above 80%. That would be standard in the clinical trial. About 30% had a power between 31 and 80%, and 57% of studies had a power of 30% or less. If a doctor would go to an ethical committee for a clinical studies saying, well, we are powered with 15 or 20%, the committee would steer him away right away because the whole trial would be unethical to begin with because it has no realistic chance of finding something that is real. This gets even worse by a practice called p-hacking. P-hacking is an umbrella term for practices where you change the experimental or analysis strategy after initial results have been observed. I see that some people are taking photos. Feel free to do that. If you contact Professor Centurion, he has the file and he's authorized to share the whole presentation with you if you so wish. Yeah, no, but I saw some people in the audience on their phones taking photos. I wasn't talking to you. Examples. You've done a study with N is 5 and found a p-value of 0.055, said, oh shit, and then you just do 2 more. Doing 2 more can be a good choice, but then the resulting statistics are meaningless. You have measured in animal studies 4 parameters. You had planned to have parameter A as your primary outcome that had a low p-value, but parameter B had a low p-value, and now you switch the paper talking mostly about parameter B. You have a post hoc decision to normalize the data, a post hoc decision to use a different statistical test, or after seeing such a p-value, you say, oh, maybe I can find an outlier, and if I remove that, my p-value will drop. Of course the p-value will drop, but that has nothing to do with science. Science means to ask a question, trying to answer the question as good as you can, but you should be neutral to whether the answer to the question is yes or no. These practices are just massaging the data until they give you the answer you like. That has nothing to do with science. This is marketing or politics. I'm not sure what is worse. So all of these practices induce bias for finding a difference if it's not there, and even if it's there, to have a completely exaggerated effect size. So my third conclusions are: small sample sizes, typical in the experimental life sciences, for pragmatic reasons, can lead to many false positives and false negatives. Intentional bias, such as p-hacking, can lead to many misleading results. And generally, p-values are always about probabilities. They are not about truth. So in clinical medicine, pressured by the authorities and by ethical committees, they've recognized this problem decades ago. And they built defenses into their clinical research. And these defenses being pre-specify your sample size, apply randomization as blinding as much as possible, and pre-specify the protocols for conduct and for data analysis before the study starts. All of that is feasible in experimental research, but done very, very rarely until now. The only thing we cannot easily change is sample sizes. If you would run or plan to run a study and your sample size calculations tells me I need 87 mice per group, and then go to Professor Centurion, and he would say to you, if he was very kind, as he always is, what did you smoke last night? But clearly he has no budget to pay for that study. And that's not an issue of Sinvestaf or Mexico, it's the same in Germany or the US. We typically don't have budgets for these big studies. But if you wanted a really robust study, in some cases we may need so many samples. So generally, there are temptations. We all are pressured to generate publications. And negative data are so much more difficult to get accepted in good journals that there is a temptation to just make the data look a little more attractive. So there are temptations to modify design elements, but this post hoc design no longer answers our question, but uses manipulated data to obtain a desired outcome. Our protections, again, and a lot of this will occur unconsciously. The smarter you are, the better you are in inventing excuses for those modifications. And if you're really smart, you can even convince Professor Centurion that these modifications are justified. Good luck. So our key protections to generate robust data are to pre-specify all relevant aspects of a study, design and analysis, and to apply blinding and randomization as much as feasible. How do you recognize when reading a paper Is this likely to be a robust finding? Well, are the authors transparently communicating what really was done with sufficient granularity to repeat it? Are they even talking about exploratory words, hypothesis testing? Are they talking about pre-specifications of their protocol? Are they talking about randomization and blinding? Do they openly discuss issues such as attrition or outlier removals? And what always makes me very skeptical as journal editor is if a paper has more p-values than sample size per group. I've seen papers, horrible papers, which had more than 100 low p-values based on a study which only 6 animals per group. That inherently cannot be a meaningful study. In the words of William Edwards Deming, it's not necessary to change, survival is not mandatory. However, all of what we are doing depends on people trusting us that we will do something meaningful with the money, whether it comes from the government, whether it comes from charities, whether it comes from a pharmaceutical company. They all trust us that we are doing something meaningful. If we don't get, as a community, I'm talking about all of us, our act together to make our research more robust, I can predict you financial support in society for what we love doing, our research, will diminish, and doing research will get much, much harder. And with that, I would like to close.
Right.
I realize I talked a little longer than I was allowed to, but if Professor Centurion allows, I'll be happy to address any questions.
Yes, please. Do you have any questions, the audience? Please. Thank you.
I would like to ask how to reconcile the use of higher number of animals with the restriction of using animals in research.
Yes, very good point. I've talked to several animal welfare officers about that, and it's an active topic of discussion in their community. And there are two arguments. One is using fewer animals is ethically good, but the other side argues if the study is underpowered, in principle, there's a big chance that all the animals are wasted, no matter how small the study is. So in several organizations, the consensus has become, let's rather focus on fewer adequately powered studies than many underpowered studies. As you saw, I worked in the Netherlands. There it's already the law. You cannot get a permit for an animal study without showing them a justification of sample sizes. In the UK, it's not the law, but the biggest funder of biomedical research in the UK is the Wellcome Trust, not the government. Shame on the UK government. And they also don't give you money for animal studies unless you can show your proposed sample sizes are adequate. So there is still no full consensus on this, but I see a trend going towards saying rather do fewer animal studies with an appropriate power than many which are underpowered. And an interim way is there are many ways how you can increase the power based on your sample size. Mathematically, if you can reduce by fine-tuning your methods and other things, if you can reduce variability by half, it's the same as increasing sample size by a factor of 4. Or more generally spoken, if you can reduce variability by x, it's the same as increasing sample size by x squared. So working on refining your techniques to reduce variability works miracles for your sample size calculation.
It's very interesting, but you almost made us cry.
I do.
Yeah, and I mean, you made us think. No, I mean, many questions, because how do we reconcile again the fact that we are a small research facility with a low amount of money, with I mean. And I have there are some things that we can easily do, like randomized studies or blind studies. These we can do easily in our lab. But there are things that we cannot do. We cannot easily have so many numbers of big numbers. No. So what's the solution?
I see the problem, and I could touch only on the surface what can be done. If you find the money to support my travel expenses, I'll be happy to come back and run a 2-day course for the students on focused solely on this. For instance, if you ask me, is every of your studies randomized and blinded? Honestly, no. But when we now do new studies, we think about what are the most vulnerable steps. And then we use our limited resources. And you may think Germany is a richer country per capita than Mexico, but still our resources also are limited. We focus our limited resources on the most vulnerable steps. For instance, something notoriously vulnerable is anything related to histology. You could say for quantification I use image J, and that's a computer that's not biased, true, but the person picking the viewing fields is. So for the last 5 years running, every study we did involving histology, the person picking the viewing fields had no clue which sample belonged to which group. So these are doable steps. I don't expect anybody from tomorrow morning doing anything perfect. Nothing is perfect, other than my wife, of course. But that's not an excuse for not trying to improve the steps we can try to improve. So bringing down reproducibility, bringing it up to 100%, rather than the less than 50%, I'm not sure that's a realistic goal. But bringing it from 40 to 60 or 70% will be a major step forward. So as a pragmatic person, I say, let's try to do the things we can do. The world is an imperfect place, unless, in case you haven't noticed.
Thank you.
Is there another question? Yes. Professor Bill.
Thank you. Hi, Martin. Well, all times I've seen you at international meetings, and this is the time I see you in my own country. Welcome to Mexico. It's kind of surrealistic for me, at least, to see you here, but I'm very happy that you're here. Welcome. I just have one short question regarding the consideration of chronobiology and chronopharmacology to explain the reproducibility of data that has been found in these results.
Okay. There could be many aspects that influence that things are not reproducible. But as I said, if a paper, so in this Bayer study, they actually, and Amgen did a similar report 2 years later. And the Amgen people talked in more detail about that, because the journal they use has less length restrictions than Nature reviews. And they said, for all of these cases, we went back to the original investigators, talked to them for all the details. In some cases, went back to redo the study at our expense inside their lab, and we still couldn't reproduce it. So yes, there are many factors such as chronobiology, strain of the animals, sex of the animals, cyclus stage in female animals. They all could contribute. But apparently that's not the big issue. The big issue seems to be the biases and the poor statistics. That's where where the money is, so to speak. Oh, by the way, which I forgot to mention, with this organization PASP, we developed an online course to train yourself on this, and some of these students from Sinvestav actually were beta testers in this course. This course is, I'll be happy to send you the link. This course is freely available to everybody. The only thing, if you want to have a certificate at the end of the course, we charge a small administration fee, because that costs us time and money to issue the certificate. But the course itself, all modules combined are about 11 hours. So 1.5 working days. And it's designed with beta testing from students and faculty from universities here at Sinvestav, participated in the US, in Italy, in the UK, in Germany, in, yes, a lot of countries. And the course is, I'll send you the link, and it's free to do. Only if you want the certificate, you must pay a small administrative fee. I think it's about 200 pesos. But the course itself is completely free, and you can make that mandatory for all of your students and junior researchers.
Is there another questions, please? Okay, right. Thank you very much, Martin. You can always talk about with him about the science and the reproducibility problems. In fact, we have, as a researchers, we need to put some data in some databases. So sometimes the journals ask about the data. You need to publish the original data. Sometimes we did, sometimes we don't. So we think that we need to do it, to share all the data we are producing. Okay, thank you very much.